my group for talking today. And hopefully they'll each go through 10 minutes or so of cool stories on the specific corner of our lab where we try to figure out how electrons cross membranes. I'm going to switch here to screen sharing and show you. So what we normally get at seminars when we present are a lot of people who are from a medical field. We certainly get a lot of questions about electrons. Usually the question is, who cares? But today, hopefully, we've gathered a bunch of people who really care about electrons moving through the environment, how they move to rocks and minerals, and hopefully everyone here understands that life is pretty much electrons looking for a place to go. We have a large group. We're only going to meet a couple of them today, but we're all focused one way or another on the microbiology, electrochemistry, engineering of devices that study electron flow to and from bacteria. So this is really the moment everybody has when they start studying iron, is they realize there's no such thing as iron. There's multiple forms and oxides. There's manganese forms and oxides. The pH matters. The crystallinity matters. And this isn't just important from the standpoint of solubility, because this makes most of the interesting forms of iron inaccessible to microbes unless they can move an electron across an insulating barrier of their membrane. But this also changes the character of the electron acceptor from the standpoint of its charge and from the standpoint of the amount of energy that it offers to a microorganism. This is a figure photograph from a recent textbook that I really like, because it shows the continuum of energy that's available to organisms using perhaps the iron that we use in our laboratory. We often make ferrihydrite, but we have a lot of phosphate in our medium. A lot of the most abundant minerals in subsurface environments have a very low redox potential, but then as you go out to the more freshly precipitated forms and as you get into the manganese forms, the amount of energy changes radically. No one would ever say, I don't know, I did my experiments at pH 6, pH 9, who really cares? That's what you're saying if you are ignoring the PE, the electron concentration of the environment. No one would ever ignore pH, and we don't think you should ignore PE. And so we're really trying to study how microorganisms move electrons to surfaces, taking into consideration their charge and the amount of energy available. So, as we said, iron's a pain. There's a lot of different forms of iron in the world, and as soon as you put them in a test tube, as soon as you autoclave them, as soon as you add phosphate or other minerals, they change. As soon as the bacteria start to reduce them, the accumulation of the other the products change the redox potential and character. And so one thing you are always asking is, there's got to be a better way to do this. And we have started to use electrodes as a way to study this, primarily because it's a way to control it. And there's a lot of other reasons to study, to use electrodes in the study of electron transfer. But today we're largely going to talk about using them to control microbial growth. These are some pictures of our most recent chambers. They are basically ways of containing 10 to 100 milliliters of anaerobic water medium in a chamber with an electrode that's going to be poised at a redox potential that we control, that's going to be kept at a temperature that we control, it's going to be stirred, it's going to have a headspace free of oxygen, and hopefully this is going to simulate some environment that we care about. And this has been the product of about 10 years of trial and error. Each new student usually comes in and asks why we do something, and we say it's the way we've always done it, and they change it, and it gets better every year. So when you're building one of these devices, you really have to kind of decide what questions you want to ask. We are microbiologists, and if we're studying electron transfer, we are kind of moving towards electrochemistry. And so what we're going to talk about is how to stay right at that interface, but we're really trying to ask microbiology questions. When you talk about scaling these things up to a fuel cell or to some other kind of device, you're engineering the system. Maybe you want it huge. Maybe you want to make an amp of current. Maybe you want to make a fuel. At that point, you're taking the catalyst of the microbe, you're hooking it up to some kind of electrochemical device, and then engineering it to work the way you want. Inevitably, when you do that, you're going to make compromises. You're going to add variables. And so we try, when we're studying the bacteria, if we want a mutant phenotype, we're trying to move as close to the microbiology side as possible. This means making certain decisions that you wouldn't make as an electrochemist, or building it in a way you might not build it as an electrochemist, because really, we're biologists, right? And anytime someone says, "Why do I think in terms of standard hydrogen? Why don't I use certain electrodes that electrochemists use?" I say, "Damn it, I'm a doctor. I'm not an electrochemist." And so everything you're going to see today is largely designed to achieve a steady state, so that there's a stream of electrons flowing from microorganisms at a known potential for an amount of time where we think they've reached some kind of steady state. To get all this to work, these are the kinds of decisions that we've made over the years. First of all, you want small electrodes, because you want the surface area relative to the volume of the device to be such that they don't run out of nutrients. The buffer doesn't change. It's well mixed. We're delivering everything to them, so you get this moment of exponential growth, just like you do in a test tube. We polish the electrodes so that a square centimeter is a square centimeter. If you polish with 400 grit sandpaper, you're making grooves that are 10 to 20 microns deep. Those are places for bacteria to hide. That doubles or triples the surface area of the electrode, and you can't compare your data with other people. Even the sandpaper that you choose can make a difference. Many sandpapers have binders and coatings that is going to leave something behind on the electrode, and we try to sonicate them and clean them so that they're the same every time, and we pitch them after use if there's any reason to believe they have something bound to them, like flavins or other compounds that are in the medium. We calibrate our reference electrodes. Everybody's got different reference electrodes. Everyone uses different silver, saturated solutions, mercury. I don't care, right? I'm a biologist. We calibrate all our electrodes versus a common master, and we report our data versus standard hydrogen because, number one, that way we can all compare data, and number two, because that's the language of biology and geochemistry. Electrochemists may use really large counter electrodes. As the electrodes flow out of the bacteria to this working electrode, we count them and we push them back into the chamber. If we were doing some sort of really fast electrochemical method where we were switching potentials, we might need a really large counter electrode to get those electrons back in. But if you have something, again, like flavins or redox active molecules, that large counter electrode can reduce that, or can reduce other compounds in the medium. We have a small counter electrode, so hopefully it fizzes off a little bit of hydrogen, and that's the only other thing we have to worry about. We blow that off by gassing the headspace. You should never have air quotes around anaerobic, just like you should never have air quotes around sterile. If you build a chamber and you don't seal it well, even if you're flushing it, our measurements have shown about 100 part per million oxygen will be in that headspace. If you use a large inoculum and you have a facultative organism, they'll strip that out. But this is anaerobic microbiology without reductants, without redox buffers, and so you really do have to work in the most strict anaerobic conditions you can imagine. And again, we have every year kind of added something to it that's knocked another 10, 20 parts per million out of the headspace, but it's extremely difficult to keep the oxygen out of these. We respect evolution, we fear evolution. If you inoculate something in a chamber where there's food in the form of an electrode surface and it doesn't grow for a week, that is a week of incredible pressure for an evolutionary event, for the sticky one to arise and grab hold. And so we like experiments where you inoculate, you get an instant result, and in 2 or 3 days you see what the organism can do, and you break it down, autoclave it, and do it again. Long-term experiments, especially with biofilms, are basically evolution experiments. Everyone has a different size and shape electrode. Current per electrode is irrelevant. Every electrode has to be harvested, and we have to count the amount of biomass on that electrode. So we are looking at respiration rate per unit cell, just like a regular microbiology or biochemistry experiment. We're not engineers trying to make the most amperage or microbiologists. And finally, be aware, all these potentiostats have a lot of buttons on them. And everybody, ourselves included, likes to push those buttons. Pushing buttons is not the same as asking questions. And when you start sweeping the potential really fast or pulsing electrons into these chambers, you will get data. But you really have to calibrate that against what you know about the organism. Verify you're not killing them. And really try to figure out why we're using these devices, what questions we're asking. And typically what we're going to talk about today is we're asking if electrons can flow out and at what potential. And so here's a little video we made for a recent program review that just shows how fun it is to do this in the lab. Basically what we start off with is this little chamber, it's about 10 milliliters, and an electrode, and this is a biofilm of geobacter, about 10 to 20 microns thick. So here's our little chamber, just set up on the lab bench. and we've starved it. We've taken away all the acetate so that it's not making any current. You can see this real-time trace down near a flat line. It's basically not making very much electricity because the bacterium has no electron donor, there's nothing to strip electrons from, and so there's no current. So what we just do is walk up, we take a syringe, and we shoot in a little bit of acetate, 10 millimolar, and as fast as you can sort of watch it mix around in there, you notice it's not turbid. Electrons are only coming from the bacteria on the surface of the electrode. Within 5 seconds, as soon as you can turn around and look at the system, bam, electrons come screaming out of this organism. And this is a miracle. We've got the entire TCA cycle running. We've got an entire electron transfer chain running. We're crossing insulating barriers, and in real time, you're watching electrons come out 5 times faster in 5-10 seconds. This is something that never gets old. It's kind of the, my God, it's full of stars moment for most students, I think, when they see it happen. Because this is the miracle of microbial electrochemistry, and you can do it every day. If you stick around for for a few minutes, you'll see them adapt, start turning things on, ramping up. But in general, this is what you can get, half a milliamp of current from a biofilm 10 microns thick, and it can turn on and off at this scale. So if you're doing it right, you can have tons of fun. So now I just want to walk through kind of what we consider to be the ideal experiment. The ideal experiment where we know that what's happening in the chamber is microbial physiology in action. Bacteria doing exactly what bacteria can do. Electricity is not magic. It's not like there's such thing as electricogens. These bacteria exist to respire, make ATP, just like every other organism, and so they should do it they should do it on electrodes just like they would do it with metals. First thing is, when you inoculate these chambers, we want to see rapid attachment colonization. This means they're hungry. They want to do this. They want to get rid of electrons. Within hours, we see a small inoculum colonize the electrode, and you see a very rapid increase in current from nothing. And you see within a few hours, that's already starting to turn into an exponential increase. The phase of growth the bacteria are in when you inoculate them is going to matter. Geobacter traditionally likes to be in a acceptor-limited state late in its growth curve. Shewanella, if you use it at that point, they're dead. They basically won't touch the electrode. The material matters. Shewanella hates gold with a passion, will not touch it. Geobacter doesn't mind. And when you're going from one material to another, if you want to use a new material for a new kind of device, we put an old electrode in there, like graphite that we know works. We put another one in that chamber side by side. And if we can't get the same kind of data from both materials, we start to worry. The redox potential not only changes the energy available to the bugs, but changes the charge of the surface. And we can drive bacteria off of surfaces just with a redox potential. So this event is so important that it was the thing that got us really excited when we first started looking at mutants. We found a lot of mutants that compared to wild type, which again, within hours, is off the charts growing. We found a lot of mutants which, as soon as we would throw them in these chambers, flatline for days. And these fail to make electricity, yes, but what they really do is fail to even show up. They fail to attach. And so the failure to attach has nothing to do with electron transfer. These are mutants in outer membrane cytochromes, which obviously change the outer surface. These are mutants in polysaccharides that are exported to the outside. Many of the global pili mutants even are really bad at binding to surfaces. If you wait 2 or 3 weeks, yes, you will accumulate a layer of cells on there. But in these experiments where you ask, can they do it, the answer is no. But it's not because of electron transfer. The next phase of growth we really like to see is an exponential phase that is when there's excess electrode surface. So every cell now is respiring happily. They make another copy of themselves, and they make the decision to stay. And so you see an exponential increase with Geobacter. It's as fast as with any other substrate we use, which shows they're very happy with the situation. This is the time to actually measure how fast they can respire. Even though this is when they're making the least amount of current, this is the moment when they have everything in excess. So the respiration rate per unit cell, this is what they're capable of. Everything downstream from here is going to have limitations. So this is really the phenotype of an organism before they start running out. As they begin to layer onto this electrode and run out of area, the next decision is if they can stick to each other and grow away from that electrode using mediators or conductive pathways. This is where the growth rate may change. you may start to see them slow down, because now you're not studying electron transfer as much as you're studying electron transfer plus something else. We have a lot of mutants, Caleb's going to talk about one of them, that fail to make it past this stage. And this tells us that the electron transfer event out of the membrane requires one set of proteins, and then attaching to other cells and building these longer range pathways is a completely different adaptation. And then finally, if you're able to attach to each other and form these layers, you may get in the realm of conductivity. And conductivity is a difficult thing to study because it requires all these other things to happen perfectly. And if you're going to make a mutant in conductivity, you have to establish you haven't broken any of these other events. This is what makes Geobacter and some other organisms able to make these incredible amounts of electricity on the order of a milliamp per square centimeter. This is as good as any enzyme-based system ever developed. And conductivity is a can of worms. And the only thing I can say about it is there are a million ways to study it. It can be a physics question where you apply a voltage across a gap. This voltage may be, in the language of redox potential, 5 volts and 5.5 volts on either side of a gap. We have no way of calibrating it when you're just doing voltage gaps. There's other experiments where you're able to control the redox potential, and that way you might be saying there's 0 versus standard hydrogen on one side and 200 millivolts versus standard hydrogen on the other side. And all I can say is we're still working out how we even do these experiments and which ones are kind of telling us about the properties of molecules and which ones are telling us about the kind of what's happening under biological conditions. But that is a seminar for another day and week and month. So I'm going to hand it off now to Caleb, who's going to tell you specific stories about a few mutants. And while we're doing that, I will listen and or watch the Twitters to see if anyone's got any questions about how we build these things.
Go ahead and fire questions to micro seminars.
Anyone?
Nothing right now.
Just a lot of thumbs up. All right, we'll go ahead and, yeah, I didn't see anything pop through. We got it live up here. So go ahead and introduce yourself. Take it.
All right, so my name's Caleb Levar. I'm just one of the resident home brewers in the Bond Lab, which ends up working out pretty well for everybody else. And what I'm going to be doing today is telling you a little bit about some work with mutants that I've done and expand on some of the ideas that Daniel mentioned about what we are looking for in a good electrode experiment and what we can learn about a more environmentally relevant electron acceptor, something like an iron oxide, from those electrode experiments. So to do that, I'll mention briefly 2 different mutants that I've worked on, working sort of from the outside of the cell, the extracellular matrix and the outer membrane, down into the inside of the cell. So the first mutant that I'll mention is a mutant in a type 2 secretion system pathway. So this is a protein complex required for moving some folded proteins from the periplasm to the outside of the cell, whether it might go back to the outer membrane or perhaps get secreted into some extracellular space. We don't know for sure. In other dissimilatory metal-reducing organisms like Shewanella, we know that type 2 secretion is essential for metal oxide reduction because it exports this C-type cytochromes that are needed to get electrons past that outer membrane. And so as Daniel mentioned, one of the things that we look for in a good electrode experiment is rapid current increase after inoculation. And that tells us that it's a mutant then that we might want to get interested in studying. And so what we're looking at here is current increase on the y-axis over time on the x. And when we inoculate both wild type and this mutant into this bioreactor, what we see is a very rapid increase in current, like Daniel was showing you. And importantly, this mutant seems to have no problem reducing that electrode. If anything, it outperforms the wild type during this initial phase of growth. And what's important to understand about this phase of growth is that the electrode surface is not limiting yet. So up to this point of about 24 hours when we're making somewhere less than 75 microamps per square centimeter, there still is exposed surface area that new cells can attach to. After that point, however, is when things get a little more interesting. So what we see as we carry this experiment out further is that the wild type continues to increase in its rate of respiration on that electrode, whereas the mutant plateaus and really fails to get much above that 75 microamp per square centimeter rate. And what that tells us is that something about running out of electrode surface area here is changing the way that this strain is interacting with that electrode. So if we make the assumption that the electrode surface is completely covered, there's really two possible explanations for this phenotype here. The first is that cells are doubling and new cells are being made, and that cells are remaining attached to that monolayer of electrode-attached cells, but failing to contribute to productive current increase. The other hypothesis is that cells are dividing and simply being released into the planktonic medium. So to answer that question, we can turn to confocal imaging, and what we're looking at here are side projections of biofilms growing on an electrode. So these green masses up on top are live dead stained biofilms, and down here where the text is would be the electrode. Scale bars in all cases are 10 microns. And early biofilms, right around this 24-hour time point, look essentially identical for the wild type and the mutant. We have a nice layer of cells. As we carry that experiment out further, where we start seeing this difference in current production, what we see is that for the wild type, we have a thick biofilm where there's obviously cell-cell connections happening, but for the mutant, all we have is still this very similar layer of green cells down here on the electrode. And so what this indicates to us is that cells are being released into that planktonic medium, and we can actually measure the release of those daughter cells directly using optical density measurements. And so this begins to break the biofilm, the development of the biofilm, up into discrete steps requiring different protein components. So moving inward into the cell now, another mutant that I've been working on is a C-type cytochrome that we've taken to calling IMCH, for intermembrane cytochrome H. And we get excited about a protein like this because it looks like it could be a intermembrane quinone oxioreductase, so a protein needed for getting electrons out of the intermembrane and into the periplasm, where they could then be moved out to the outside of the cell. And one of the hypotheses that we have about mutations in such a protein is that it should really wipe out all electron transfer. And sure enough, when we inoculate those cells into these bioreactors, what we see is a flat line in current. So again, what we're looking at is increasing current over time with wild type in black, and we see this flat line. Now Daniel told us to get worried about a mutation that yields a phenotype like this. So it's important to note here that these cells remain able to attach to the electrode. So when we look at very early electrodes and we harvest them and measure the amount of attached protein, we still have a fair bit of protein attached, and perhaps even more than you might expect because there's so little current being produced. So there's cells attached, they're just not reducing that electrode. And so really what this all comes down to is that biofilms require distinct events to form this thick biofilm with cell-cell attachment and cell-cell electron transfer, like we think about for wild type. And you'll notice the data looks a little different here because it's shown on log scale because we're microbiologists, and that's what we do, right, Cameron? And so what we're looking at then is just the same data. We've got this IMCH mutant down here on the bottom, which attaches only, but we really don't have any appreciable electron transfer to the electrode. We have then this mutant in type 2 secretion, which is able to attach to and respire that electrode, but fails to increase in current past that monolayer stage. So with that in mind, I want to take a step back quickly and talk about iron oxide and what these types of experiments may or may not tell us about iron forms in the environment. I got interested in this stuff because I really wanted to learn how what we learned in the lab might translate to what's going on underneath our feet in northern Minnesota. And you might have gathered from some of Daniel's earlier slides where you have Angry Bear asking how many forms of iron there are, that it's a really complicated situation. Fortunately for Geobacter, we can narrow that down a little bit and really focus just on ferrihydrite and goethite as the primary iron oxides, but that really doesn't simplify matters too much. As Daniel mentioned, we have various forms of these minerals ranging from amorphous to crystalline, and these changes are going to happen in your fridge in a hydrated sample of iron that you might want to use for an experiment a month later. They're going to happen as your bacteria are reducing that iron. Surface area to volume is changing the whole time. The redox potential is constantly changing. This is a very different situation than something like an electrode, where we have an inexhaustible electron sink held at a set potential. And we can also have a lot of other elements like phosphorus and sulfur and arsenic that are incorporated in these minerals which complicate things further. And so from these, it really becomes evident that we'd like to use something less complicated, something a bit cleaner, and use electrodes to mimic what's happening on iron. What we want to ask, though, is do electrodes really equal iron oxide? Because if they do, we should be able to make some predictions with these mutants on what iron oxide phenotype we should get. So I'm going to cheat on wild type. We're making lots of currents. Of course, it reduces iron oxide. We know that to be true. For something like this type 2 secretion mutant, where we have an intermediate current production phenotype, you might expect that it would reduce iron oxide to some extent. It's able to attach to surfaces, and it's able to get electrons to those surfaces. And for something like delta IMCH, we're not getting electrons out of the cell, so obviously we should have no reduction. Well, it turns out that's not the way it works. So wild type up here on the top, we're forming a thick conductive biofilm, and of course reducing iron. So what we're looking at here is the accumulation of the reduced form of iron in the medium over time. The mutant, which was the flat line on the electrode, unable to reduce, is able to reduce now iron oxide at a rate and to an extent similar of wild type, and the mutant that had the intermediate phenotype on the electrode is unable to reduce that iron at all. So looking at it all together, and you can just really look at colors here and see that in general the blue and red traces have flipped. So things are not the same electrode to iron. So it's really important to consider the different variables that iron and electrodes bring to the table, and to be careful about the conclusions that we're trying to draw or the correlates that we're trying to draw between them, and make sure that we're asking the questions that we think we're asking. So just to reiterate, not all surfaces are made the same, and we need to think about what questions electrodes can tell us, and what questions electrodes can answer, and what questions electrodes can't answer. But Lori now is going to tell you a bit about some of those that they can answer, and as we're switching over, I'd be happy to see if there's any questions coming in anywhere.
Appreciate the zinger.
Yeah, I had to, sorry. Well, I'm not sorry. Never mind.
Good for you. Any questions? Tweet to micro seminars. Hashtag micro seminars.
There is?
Yeah. This is still one.
So on YouTube, he's still talking?
I guess so.
Group hasn't had enough coffee yet.
Okay.
So by the time the first
Can we
question
just
comes
go?
through-
All right.
Go ahead. We'll ignore them.
All right, I'm Lori Zakharoff. I am a current graduate student, and I'm just going to continue talking about some interesting phenotypes we've seen in some of the deletion strains we've made in the Bond lab. Okay, so my project started out with the question of, can we start to study all of the intermembrane cytochromes in Geobacter, because that is essentially what is the most important step in respiration for the bacterium itself. It's what connects its metabolism with the electron acceptors on the outside of the cell. So that doesn't sound like too difficult of a project. Let's just start at looking at the genome and seeing what we have. And what you find when you just look at the genome is 5 large gene clusters that can potentially generate a proton motive force. And so that really didn't narrow the question down too far for me. So we started looking at the data that we had already in lab from mass spec and also from some transposon screens, and we got more interested in this protein, which we're calling CBCL. And why we like it is that it has a periplasmic domain with 9 heme groups in it, and it has everything you need in the inner membrane to generate a proton motive force. It has predicted B-type hemes and predicted quinone binding sites. And that's compared to all the other CBC groups there are, in which these components are separate. So we would think that if we maybe knock this out in the cell, that we would get a pretty severe phenotype. And so that was the first thing that we do in the lab. And we're a lot more excited about this now because of the ability to make markerless deletions in Geobacter. So that's what we're looking at here. And when you knock this protein out, we don't get a very exciting phenotype. And when we're monitoring reduction of insoluble iron oxides, we notice that it can do it, but just not quite as well as wild type. We give it an easier situation, that is reducing soluble ferric citrate. It can do that at near wild type levels. So that wasn't really telling us too much about an intermembrane phenotype, and if anything it says, well, maybe this isn't too important for respiration using iron. But then we're going to do the same experiments we do with everything else and grow this mutant in a poised bioreactor and see what happens. And the growth phenotype here wasn't too exciting either. It reduced, it attached to the electrode, and the cells that were there made current, just not as well as wild type. So we turned to another technique that we like to use in lab, and that is cyclic voltammetry. And the idea here is that you're going to poise the electrode at a very low potential, and that potential should be very unfavorable for electrons to move from the biofilm to the electrode. So that's why when you monitor current here, you see no current being produced at this low potential. But then we're going to slowly scan the potential higher and higher at 1 millivolt per second. And when we do that, we're going to eventually get to a point where electrons start leaving the biofilm. And as we increase the potential more, the biofilm becomes less reduced as the electrons move from the biofilm to the electrode. And when you get to the midpoint here, that's when we consider half the biofilm to be reduced at that point. And then eventually you reach a steady state where the potential is favorable and the electrons keep flowing out of the biofilm, and then we sweep that back. And the cool thing about Geobacter is that no matter what you do to it, the cyclic voltammetry always looks the same. So you can do that at a full-grown biofilm here, or even at an earlier biofilm. So this shows us that no matter what the conditions are, even if you're having cells that have no limitations and don't have to pass electrons through an entire biofilm, the cyclic voltammetry looks the same as a full-grown biofilm. And the only time we've ever seen any change to that was when we were doing the cyclic voltammetry for the delta CBCL. And what this shows us is is a positive shift in the midpoint potential of the cyclic voltammetry. And to us, that says that there needs to be a higher driving force in order for the electrons that are there to leave the biofilm. So we're scanning along here, and we pass the point of favorability for the electrons to leave the biofilm, and we have to get a little bit more positive in potential for the electrons to be pulled out of the mutant biofilm. And again, this is the only time we've ever seen this particular phenotype. And the take-home message for us has been is that there has to be some other intermembrane protein that can pass electrons from the inner metabolism to the extracellular space, but it's not as optimal as CBCL. Just to reiterate, that means that we think there's probably multiple paths involved in moving electrons from the inner membrane, and that is contributing to the kind of mixed phenotypes that we are often seeing when we're doing insoluble iron reduction and reduction on the electrode. Okay, so that is that for the story of CBCL. So I'm going to pass it over to John, and feel free to ask any questions in the meantime.
30 seconds. We should do the Google sound effects.
So I have a question actually in terms of this protein inside the membrane.
Yeah.
Hey, guys, I'm sorry to interrupt. There's a lot of feedback on that side from regardless of who's speaking. I was wondering if you could turn down your speakers a little bit and see if that helps.
Oh.
Okay. I think that's
Okay,
better.
we try it now. Does that sound better?
Yeah, that's a lot better. Okay, go ahead, Jen.
Okay, sorry, now I've got to compete with a 4-year-old. Jack, do you want to say hi?
Hi.
Can I ask my question now?
Hi.
Okay. So I had a question about that inner protein. Is that something that you think happens in other organisms also, or just Geobacter?
That is something we're very interested in. John has sequenced a few more in Geobacter, and we've been looking for proteins that are similar to Geobacter, and of the species that have been sequenced, CBCL seems to be one of the more conserved CBC proteins. And so we think, yeah, that there is something there, and that this might be the main pathway, but Geobacter interacting with more varieties of potentials might have other pathways in the inner membrane. Yep.
I have a question also on this regard. What kind of... Sorry, we're still getting a lot of feedback.
Oh, sorry. When I turn the volume down, then I can't hear you.
Okay, how about a happy medium there? That sounds good. What kind of mechanism do you think is showing this change in midpoint potential? Are you thinking that it's like another cytochrome with different potential itself, or are you thinking of like a completely different type of transfer mechanism? Or I mean, what are you guys speculating at this point?
We're thinking that it's more probably another protein, because there's a few different thermodynamic problems that could be causing this phenotype. It could be that the proton motive force is perturbed in this situation, so we need a higher driving force to overcome that, or it's a different protein that has a different potential that needs to interact with it. And it's interesting that I can poise the electrode and grow the organisms at a higher potential, and it has no difference in phenotype. So whatever this other pathway is something that it doesn't appear to be able to increase its amount of current production with.
Okay. We have a question from Twitter here from Ben Temperton. He's asking, is acetate used as a carbon source for ecological reasons, or because it's cheap and it works?
Well, yeah, we've always used acetate both for ecological reasons and yeah, sure, I suppose it's cheap, but I guess we don't really think about acetates being cheap and that's why we use it.
What about fermentation?
What about fermentation?
I'm assuming there's an additional benefit for using acetate because it's non-fermentable, right?
Yes, but whether or not, I know Geobacter doesn't perform substrate phosphorylation, probably not fermentation, so I don't think that's a concern for us.
Yeah, I think there's in some ways this is a historical thing. A lot of the anaerobes were isolated using acetate initially because they were trying to find respiratory organisms as opposed to fermentative organisms.
Yeah.
Cool. Okay. Well, why don't you guys continue?
Okay. Yeah, it's always a feedback problem, but then you can't hear the questions.
Okay, thanks everybody for tuning in. My name's John Badalamenti. I'm a postdoc here in the Bond Lab and also one of the other resident homebrewers here in the lab. And we've heard some really cool stories about how we can use electrodes to study phenotypes in Geobacter sulfur-reducens, but one of the things I'm interested in particular is taking some of the techniques we know about electrodes and applying them to new environments and hopefully using them as bait to attract new bugs from new places. And so this is a picture of me sampling some anoxic brine from about a half-mile underground at level 27 of the Sudan iron mine located in northern Minnesota. And what's really cool about this field site is that it transects a massive 2.7 billion year old banded iron formation. And these anoxic brines are just teeming with microbial life ripe for the picking. And so we think that electrodes are a really cool device to access some biodiversity in this habitat. And so I'm going to be talking about some of my work as well as previous work aimed at using electrodes as bait to bring these subsurface microbes into the lab. And so one of the approaches we've taken is pretty simple in that we start with brine taken from the mine and we use that as the inoculum for electrode bioreactors. We poise at an oxidizing potential and we wait for what sometimes can be a painfully long time. And you can see here in this current trace that it took about 30 days for us to finally see the selection of electrode respiring organisms from this brine. Now, hopefully, Caleb has convinced you that studying microbial respiration on iron is not necessarily the same as studying microbial respiration using electrodes. And so what I think is really powerful about this approach is that it allows us to ask a direct question about whether or not there are any novel electrode respiring microorganisms in this environment without first having to jump through the intermediate hoop of culturing them on iron. And one of the other cool things about this approach is that you can see here on the right, once we've enriched them on electrodes, it's then relatively clean and easy for us to harvest the biomass and proceed with further characterization, whether that be isolation or metagenomics. Another approach we've taken, and this was done primarily by our former postdoc Zara Summers, is to bring the electrodes to the mine and do some enrichments in situ. And so a lot of the data I'm going to be showing for the remainder of my slides is the culmination of a lot of the groundwork that she laid when she did her work here and handed off to me when I started. So if we fast forward from some of Zara's work through about 2 years of headaches and head scratching, I'd like you to meet our first iron-reducing isolate from the Sudan iron mine. It's a Disulfuramonas. We've named it Disulfuramonas sudanensis. And you can see here that it grows on electrodes. It has about a 1-day doubling time. And what's really striking about this isolate and really exciting is that its respiration appears to be tuned for redox potentials that are about 100 millivolts more positive than we see in Geobacter. And so what this tells us is that we think that this Disulfuramonas isolate is using iron forms in its natural habitat that are fundamentally different from those that we typically prepare in the laboratory. And now, as cool as this isolate is, one of the reasons why it took me so much time to isolate it is the fact that it grows very slowly. It takes about a week in culture tubes and 2 to 3 weeks on plates. And so doing a simple microbiological characterization, like testing different substrates as electron donors, all of a sudden becomes pretty laborious when you think about the number of tubes and the amount of time that's going to take. So one of the other cool things we can do with electrodes is to instead of that taking weeks or even months, we can do that in a matter of hours. And so this is testing this disulfur-ammonious isolate for its different substrate utilization capabilities. And you can see here that on an electrode containing attached cells in otherwise donor-free medium, we see a response when we spike in different substrates, for example, lactate and ethanol, immediately, very similar to that video we watched earlier. For pyruvate, we see a little bit of a weaker response, but for other substrates that this organism cannot metabolize, we see a flat line. And so this is a really fun, kind of quick and dirty way to answer some basic questions about the isolate and then back them up with some old school microbiology down the road. And so this is, that picture is missing, but I had a picture in here. This is what I think is most exciting about this isolate and being able to do enrichments on electrodes is that we can assemble complete genomes from the isolates once we have them. And so this is the complete closed genome of the disulfur-ammonious isolate. It's just under 4 megabases, and you can see in the green, the red highlights around the perimeter, it has 39 predicted multi-heme C-type cytochromes. And this is a really small number when we compare this isolate to other members of this family. And so we think that this organism may have converged on a minimal set of cytochromes required to perform electrode respiration and metal reduction. We also see evidence for possible arsenate reduction and some other cool things listed here as well. The blue bars show areas of potentially horizontally transferred DNA from phage. And then finally, just a kind of a plug for how we obtained this genome. This was done with about 250x coverage of just PacBio long read data. And I know that PacBio gets a little bit of a bad rap for having what is a very high error rate, but what we can do is actually computationally resolve that because that error rate is random. And with enough depth of coverage, we can actually use the long reads to resolve repeats in the genome sequence and actually get a closed circle. And so you can see here on the left, this little green highlight here is actually a tandem duplication of ribosomal RNA operon. And in the environment, we expect this organism to only have one copy, and as we domesticated it in the lab, it probably made a duplication. And using long-read sequencing, we were successfully able to put this into proper genomic context. And so I'm a huge proponent of PacBio. I know the folks around here think that I sound like a broken record, but I think it's becoming the rule, at least for microbial genomes these days, and no longer the exception. And so my plug is, if you have a cool isolate and you want its genome sequence, this isolate costs us less than $1,000, so I would highly recommend it. So with that, that's the end of our seminar today, and feel free to tweet or ask me any questions or any questions you have for the rest of us. Thanks for tuning in today.
Very cool. Thank you, guys. I really enjoyed this alternate format. I hope the rest of the people watching did too. There's been at least one question that's come on that hasn't been answered over Twitter from Mike Wilkins regarding this disulfur-ammonious isolate. He asks, can you poise the electrode at elemental sulfur and see if it can reduce that?
I don't see why not. I don't remember the midpoint potential for elemental sulfur off the top of my head, but with the potentiostat, we can poise it at whatever we want.
Cool. So, Mike, do you have a follow-up to that, or I'll give you a second. Let's see. All right. Well, I have a question for Caleb. If we can go back to, or anybody in the group actually, if we can go back to this concept of how the electrode, the one mutant seems to reduce the electrode really well, and one mutant seemed to reduce iron, and they weren't the same. You guys sort of maybe this is a teaser for future studies or work you're doing now, but I was wondering if you could give us your thoughts as to why exactly that's different. What do you think is going on there?
Well, there's a lot of possibilities what might be going on. One of the things that seems to be correlated with electrode respiration is this ability, or I'm sorry, that's correlated with iron oxide respiration is the ability to form a thick biofilm. So it's possible that something like an iron oxide is more similar, or interacting with an iron oxide is more similar to interacting with other cells, where you have to reach out and sort of grab more acceptor. So it's this idea that iron oxide is not very favorable as an electron acceptor, and you can't just touch what falls on your surface if you want to divide. You need to reach out and grab something, and grab a volume of iron oxide 2 or 3 times greater than the volume of yourself if you just want to divide once. And so there's some things going on there. There's some other hypotheses that we're working on as well, and I encourage you to keep an eye for some publications that'll be coming out from us soon as well. Or just come to ISSM in October.
Cool. I figured you guys were following up on that. Let's see. I think we've had a couple questions that you guys have answered over Twitter. Just to reiterate them for people who might follow along in the recording, there was a question about whether there's any plasmids in the desulfuromonas isolate, and the answer is no. And then there was a question about how similar the cytochromes you're tracking, the CBC family cytochromes, are to the Shewinella CYMA cytochrome, and Daniel's response over Twitter was that there's 0 similarity, and that Geobacter has a quote mess of inner membrane quinone oxidase options, unquote. Does anybody want to follow up on that?
You were right about the plasmids. And
Hey, Dan, can you guys unshare your screen so we can see your beautiful faces?
oh man, we're climbing all over. We're like pushing the computer all over the place. Okay, unshare.
That's fine. It's more exciting than the PowerPoint.
There we go. There we go.
Cool.
Yeah, no plasmids in this one. Some of the other ones that John's done have had some nice hefty plasmids, so they're not uncommon in Geobacter. The question about the CYMA, basically, if you Shewinella, the organism which turns out to have a fairly tractable iron reduction system, has a single quinone oxidase, so the electrons get out of the quinone pool, they get passed to a set of cytochromes in the outer membrane. Geobacter, as far as we can tell, has at least 6 options. And from our data, it could be using up to 3 or 4 of them at a time. And then, of course, it's got a good 20 or 30 possibilities in the outside. So it just seems to have more options, probably because it interacts with more things and is trying to wring every drop out of them. And as Caleb said, there's some aspects of this we have theories about that we're gonna kind of go on record for soon. But in other cases, it really is true. The similarities between electrodes and metals seems to be more in the fact that to share electrons with another cell, centrophically, and to share electrons with iron particles, those seem to have a lot in common in terms of putting electrons outside and letting them kind of, flow away from you. So complexity does make sense. It's just a pain in the butt.
Yeah. You guys have done some excellent work there, obviously, piecing this all together. Are there any other questions over Twitter or from anybody that's inside the Hangout?
I think we drove everyone away with technical difficulties.
there's still at least 15 people watching that I can see, and that doesn't count the YouTube viewers. So I think we haven't scared them off too badly, although this is definitely proof of our nascent phase here.
Yeah.
Well, you guys can always tweet questions to Daniel or to the various speakers that came on today. If you follow the micro seminar hashtag, you'll see a number of the graduate students also have their own Twitter account, so feel free to follow up on that. Why don't you guys go ahead and say hi?
We're going around, Caleb, for all the, put names to faces. Again, that's the lab going around the horn, and this is Lori and John. There you go. Come see us, sunny Minnesota.
Well, thanks very much, you guys. I think we'll sign off now and talk to you soon.
All right, it's been fun.
Cheers. Bye-bye.